Okay, so today's video is going to be slightly different. I'm going to be talking about the cell cycle. So basically I'm just going to teach you a bit of biology, you know, why not? Why not? The main reason is because I need to know it for my exams, but you know, why not teach you at the same time? So this is basically the cell cycle. First off at the top we have mitosis, we then have gap number 1, protein synthesis down at the bottom, and gap number 2. Now gap number 1, protein synthesis, and gap 2 are all part of interphase. This happens before mitosis begins and is kind of like the process of growing and developing of the cells, which I shall be drawing right now. So basically, the new cell organelles are synthesized and DNA replication can take place. Here are some cells and the DNA, just slightly condensing, not properly condensing, but slightly condensing. Okay, so now we're going to be talking about mitosis, you know, the bit at the top that was in the cycle originally. So mitosis is basically the splitting of cells to get 2 exact copies of the original cell, so basically 2 daughter cells. Yeah, there we go, I've written it. Oh, and here's, oh, there we go, there's 2 of me now. And this is through the process of division. So the first part of mitosis and the next phase following interphase is prophase. Now this is where the chromosomes condense and become shorter and thicker. I'm drawing spindles because they are formed during this section. They're also known as the poles of the cell. So here we're drawing the condensed chromosomes joined together by their centromere, and then the nuclear envelope breaks down, and that is the end of prophase and the beginning of metaphase. There we go, metaphase. Awesome. So the chromosomes line up at the equator, or the centre line of the cell. There we go, they're all perfectly lining up there. The spindles lengthen and attach themselves to the centromeres on each chromosome. There we go. Which leads us on to Anaphase. This is where the centromeres split. There we go. The spindle fibres then shorten and pull one chromatid, or half of a chromosome pair, to either end of the cell or the poles. They're supposed to be chromosomes, but don't really look that great. Sorry about that. Yeah, there we go. And then at the end of this, spindles break down. So the last part of the mitosis is telophase. The chromosomes unravel and the nuclear envelope basically reforms. Now these cells are supposed to be exactly the same, which is why I was trying to get them exactly the same, because they're supposed to be exact copies of each other. So please just don't judge me on that. So yes, the nuclear envelope reforms and encloses 2 sets of genetic information within separate nuclei. There we go. And then each of these separate nuclei pull away from each other, making the cell split into 2 separate daughter cells. And that is the end of mitosis. So basically, in summary, we have interphase first. That's the protein synthesis and DNA replication. Then we have chromosomes condense, spindles form, and the nuclear envelope breaks down. The chromosomes line up at the equator, the spindle fibres lengthen and attach to the centromeres split, Spindle fibres shorten and pull chromatid to either end of the poles, the spindles break down, the chromosomes unravel, the nuclear envelope reforms, and 2 separate nuclei are created. So thank you very much for watching this slightly different video. I hope you've learned something. I know that I now know the cell cycle, and I hope you could say the same thing too. So feel free to subscribe and click the like button, and I'll hopefully see you next time. Bye!